you have figured out very quickly that the cholera in Haiti came from South Asia. Tell me about that.
Right. Using the Pacific Biosciences third-generation DNA sequencing technology, we took samples from Haiti for cholera, samples from Latin America, and samples from South Asia, completely sequenced the DNA of those different bugs, and then could unambiguously resolve exactly where that Haitian strain was derived from, and we pinpointed it to a South Asian strain.
Now, some people say, what difference does it make where it came from? What has to happen here is we've seen all these people sickened and 2,000 people die. Why is it important to know where it came from?
Right. It's not just important to know where it came from, but what's the structure of the bug? You know, what are the types of mutations it carries that could affect treatment? Is it more virulent? So you want to be more aggressive in how you treat it. And not only that, but how do you prevent the spread? So it's definitely not about laying blame. It's about getting the best treatments and preventing spread that we can. You and I know that if you are sick and step onto an airplane, right, that you can be across on another continent within a day and giving that sickness to others. So the same sort of thing could have happened with the South Asian-derived strain in Haiti. It could have been somebody from South Asia going to West Africa, and then from West Africa to Haiti. We don't know. It's a global problem. The aim is how best to treat.
You haven't got patient zero yet, but here's the interesting thing, because normally we talked a year ago almost when this earthquake happened about the fear of cholera. Normally we associate it with brackish water or water which is not cleaned and that is used communally. But in this case, if it came from South Asia, then it lends to the hypothesis that somebody brought it over. It came on a plane.
That's exactly right. And if somebody brought it over from a plane to Haiti, then the same type of, then it could spread from Haiti to other areas. So one of the main aims, one of the main things being proposed is, should we be aggressively, say, vaccinating people in Haiti to prevent it from spreading throughout Latin America, or certainly to Haiti's neighbors?
What's the advance in the technology that you actually use to sequence it? Because one of the things that struck me as a non-scientist is it was very fast.
That's right. So the two things that matter most in this type of problem for infectious diseases are speed, the speed with which you can generate that data, and then the speed with which you can analyze and interpret the data. What the Pacific Biosciences technology provides for the first time ever is the ability to take an entire genome of a bacteria, same day, within just a matter of hours, get to the complete genome sequence, but not only generate that data quickly, but generate it in such a way that it's easy to analyze. These are like putting puzzles together, these problems we're trying to solve. And if you have very big puzzle pieces, then that problem's much easier to solve. So not only does the PacBio technology generate the data quickly, but big puzzle pieces, not tiny puzzle pieces that would be difficult to assemble.